Good night, good evening, wherever you are. Hope everybody is having a good Sunday. For us in the eastern part of the world, it is almost actually Monday, but the 1 a.m. But I am sure many people in Europe and in America, they are still enjoying the Sunday. So I just wanted to do a talk about my ongoing project regarding mushrooms and ecology and the Ida Mountains. And I've been doing literature search on various topics, actually, on the growth of mushrooms. And it's very interesting. And, the source of nutrients, actually. And to start off, my master work, my master's thesis was on growth of baker's yeast on low nitrogen media, and it is due to several factors, because nitrogen is one of the most crucial elements in life. I mean, like, if you look at the abundance of elements in a cell, of course it's called organic chemistry, and mostly that is carbon, because it can make four bonds, and this makes it a optimal element. For life, it can bind to many things at once. It's very interesting for carbon to be able to make four bonds. Like, for instance, methane gas, just one carbon in center, and there's four hydrogen, CH4. Just basic. Whereas, like, the oxygen, it just makes two bonds, like H2O. Because of the location on the periodic table. There is this rule called octet rule, and the valence electrons needs to be completed to 8, and so on. So there are specific chemical rules, but at the end it comes down to available electrons that you can use to connect and make bonds with, making bonds. That's what matters in organic chemistry, and that is the chemistry of life. And then there is first things first, carbon. I mean, we respect that, but on the other hand, there are other things, like nitrogen and phosphorus and oxygen, hydrogen, and which are actually most abundant elements in the universe. I mean, so when if we go back to the beginning, so nitrogen, as you see, is among the first 6 elements that we need for life. Nitrogen is very important. So there has been many research completed on the functions and roles of nitrogen, and how it is regulated and how it is used in the cell, because it is required for the growth. As I mentioned, if you want to grow, you need elements, minerals. You need material to grow, and this material usually means carbon source, nitrogen source, phosphorus, and there are trace elements. I'm not gonna go into that. Yes, life requires many trace elements. For instance, copper. Many of the fungal enzymes are dependent on copper and iron, to be able to do their, enzymatic work or enzymatic functions. Especially copper is very important for white rot fungi. And today, actually, I'm gonna talk about nitrogen, the need for nitrogen in mushrooms, and and how it is how it is addressed. How is this addressed in the living world? How organic, how life on this planet gets nitrogen. You know, in the deserts and in the tundra and cold climates and in temperate forests and even in oceans. You think oceans are fertile? No, they are not. Again, talking about nitrogen. Back in the day, we had many whales. Remember the Moby Dick, the Ishmael, the harpoonist, on the ship of the Captain Ahab. They were going on whale hunts. That, because back in the day they were using the spermaceti whales, the sperm whales, for various things, even for dressing. They would use the bones and then the fat. The head is full of fat, I guess, to counter all that pressure, oceanic pressure. Like they're divers. I mean, they dive just deep to get the cichlids and octopus and etc. Especially cichlids. The nautilus and all these weird oceanic creatures. But whales dive. We know that. And then sperm whales, have this oil in their heads full of oil and etc. So in the ocean, they do a typical job. Actually, they fertilize the ocean. I mean, the sperm whales, and normally when they are hunting, they don't, because they are in a adrenal mode, but when they are on the surface, that's what they do. The whales, I mean, the dolphins, and etc. It's part of their job. I mean, to fertilize the oceans, so you get some nitrogen in the system. In the springtime, they would migrate to northern waters, the polar regions. When the ice is melting, they would travel there and then feed on the krill and crustaceans and all the other good stuff in the northern seas. And they're very precise when it comes to navigation. They have very precision guided navigation systems. They have this amazing navigation systems, like on a route. I mean, it's just amazing how they travel all these long distances. Imagine crossing Atlantic Ocean, from one end to the other, all the way from south to north. The majestic creatures, and some of them live almost 200 years. I mean, the whales are majestic creatures, and ocean is something else. We don't know nothing about ocean. I am sorry, but that just like mushrooms, we know nothing about oceans, and yes, now we have this giant, patch of marine debris and plastic garbage floating instead of whales in the middle of oceans. They're trying to clean up because our rivers are constantly pumping plastic pollution into the oceans and into the seas. So instead of nitrogen, and instead of whales swimming in the ocean, we have garbage and garbage islands swimming. So that's a whole different. So there are some institutions, there are some non-profit organizations doing as much as they can. One is, I think, Sea Shepherd. and I try to follow their work, and they both try to prevent the whale hunt. At the same time, they're cleaning up the marine debris, especially in the Mediterranean, because all these nets and fishing nets and all that garbage. And I'm not talking about like basic, river pollution, like Coke bottles and etc. So soda bottles and etc. But there's a significant amount of, marine debris left by the fishermen as well. All these lost ghost nets and etc. So while because it is important. I mean, I was gonna talk about the fungi and the forest. See, I'm still stuck in the ocean. Anyways, I will. That's my ADHD talk. That I have to like concentrate and to get going to where I have to go. But I mean the first things first. I mean, that's why I said that the ocean is like salty water, yes, but in the middle of ocean. I mean not the pelagic zone. The coastal zones are full of life, but in the open sea, you know, there's not much. There's not much. So same thing with the with the wood, because it's just wood is devoid of nitrogen. It's just the full of cellulose, xylem tissue, and which is not nitrogen rich. Now, we're talking about a forest, all these giant trees. Imagine a redwood forest, Why we have cut down almost all of the redwood forest? Because it was fungal resistant. In the sequoia trees, back in the day, in Middle East, the civilization started on cedar. In Europe, the civilization started on oak trees. Giant oaks were cut down to build ships and etc. Same thing for the Mediterranean. The cedar, Cedrus libani, the famous, notorious cedar tree of the Mediterranean, gave rise to the all the nations and civilizations, Hittites and Egyptians and all this boat building. The Phoenicians, and they built their boats, with the cedar, and even in Bible and other religious texts, it talks about how Solomon, ordered Phoenician kings so for some timber and to build the temples and or ships and etc. It was an important source in Lebanon, today's Lebanon, the hometown of the cedar, the Mediterranean cedar tree. So because the tree was useful, for and then maybe it was resistant to the fungal infestation or fungal growth, because of the resin or other things. So maybe they were treating, with something else, but it was preferred. In Middle East, it was cedar. In Europe, it was oak, the hardest wood. In Europe, I'm sorry, in America, the sequoias, the redwood forests were used. I mean, for timber. I mean, they were like giant trees. I mean, they fell them. It's just huge, like 300 feet tall. I'm talking about. It's like a miracle, It's just like them trees that lived like 4,000 years, some of them maybe old. They were as old as like 4,000 years, and maybe youngest was like 2,000 years. I mean, they were like giant. They were like giants. The redwood. Again, it comes down to this, But yeah, yes, in the evolutionary time scale, there was this conifer age, before the flowers, before the the start of insects and etc. Even the dinosaurs were like Jurassic Park, like it ended 60 million years ago, but we're talking about conifer forests, and these are really early trees in the plant evolution. Conifers are like dinosaurs. I mean, they are primitive. I mean, it's just that their flowers. They don't have flowers actually. They're called the gymnosperms. So it's a different terminology, meaning they don't have the fruit. It's just open seed. Mean gymnosperm means open seed. It's just nothing covers the seed. As you see, it's just the pine cones. That's not a seed. That's just a wooded structure. I mean, and then again, I'm gonna talk about pine needles and pine cones and etc. Because again, we're talking about nitrogen still, even in those early times of the year, planet, you know, 500 million years ago. Imagine, imagine a volcanic eruption, just they formed a new land, or some catastrophic event, cleared up some land, and then now there is new growth. The carbon is easy to get; it's in the atmosphere, 1% or whatever. Nitrogen, yes, it is in the atmosphere, as you know, like 70% or so atmosphere is nitrogen, N2 though, not usable to any life form. Actually, to most of the life form. Well, I'm gonna talk about that nitrogen fixation. The atmosphere is full of nitrogen, the good stuff for all life. But is it available? It's just and to be honest, I don't know how we got that much nitrogen in the atmosphere. I mean, like for my colleagues who are studying planet formation, please investigate how did we get 70% nitrogen in the atmosphere. Because if you're gonna colonize Mars, for God's sake, if you're gonna take nitrogen bacteria there, if there's no nitrogen in the atmosphere, good luck. How, nothing will grow, because I'm gonna the main talk. The main topic of this talk is actually nitrogen bacteria, nitrogen fixing bacteria, because all these years, like starting from the eukaryotic life started, like, I think around 700 million years ago. It was before that it was all bacteria, and then sometime 800 million years ago, some bacteria decided, okay, we are all by ourselves and we're not doing much. Let's come together and let's come together and make a symbiosis. This is the topic of this talk, actually. The subject of this talk is actually symbiosis of the bacteria, because that's how it started. So somewhere around 800 million years ago, the bacteria, it's a cyanobacteria, gram-positive, negative, whatever, they were living happily in the oceans and in the lakes and etc. And then they said, oh, like, this isn't working, and it's been 3 billion years, and we haven't accomplished much, let's come together, let's let's do something together, let's get married, just example, just for the fun of it. The first eukaryotic cell formed. We have no idea how it formed. There are, yes, that basic theories. Looks like one got into the other. It's just it was a cytoplasmic merge. Maybe one bacteria was bigger and the other was smaller, and then they just merged, and started living together. It's a symbiosis. And then it went on because it worked. Like I said, until then life was all bacterial, and it just was in the seas and in the lakes. Nothing much happened. And then we see the rise of the eukaryotic life 70, 70, 700 million years ago, 800 million years ago. I mean, check with the chronology. That is the start of the eukaryotic life. I mean, that's when you start to see the worms and all the other like sea creatures, apart from bacteria, like jellyfish kind of life, cnidarians and then see other higher algae groups and etc. There is photosynthetic bacteria, but plant is something else. Plant grows on land, and then plant grows with fungi. Plants grow with nitrogen fixing bacteria. That is the idea, because in land it is something else. In sea, yes, it's a soup. That's what all the theories are saying. That the seas, especially shallow seas, were full of materials, organic materials, and nitrogen. Even in the shallow waters, it was present. That's how they hypothesized or theorized. You know, on the beginning of life. So but, how do you grow in an environment devoid of nitrogen? How do you grow in an environment that lacks nitrogen? You either need to get it from the air, or you either need to have better root systems, maybe like the dust associations with fungi, plant fungi associations, or some bugs, ingested these bacteria into their gut systems, in today's world. That's where we find the nitrogen fixing bacteria. This bacteria, has this enzyme called nitrogenase, and it fixes the atmospheric nitrogen into ammonia. It reduces it, the atmospheric nitrogen, N2. It has no nitrogen in it. I'm sorry, it has no hydrogen attached to it. But when you reduce it, it is an energy actually consuming process. You need energy to reduce the nitrogen in the atmosphere. But when you do it, that ammonia is very valuable, because even the proteins, like we call it amino acids, that's the amine group, is the nitrogen group. All the amino acids have one group, that's carboxyl group. That's the acid. We call it amino acid for a reason. Because it's constant. One end is the carboxyl group. The other end is amine group. And that's how and then this is the basic structure of the amino acid. Like we have 20, 25 or more, that is used in the living systems, and that there are some that is not used but utilized, as a nutritional source, or other things, like theanine, for example. It's not in the protein, but it is used as a neurotransmitter or something else. To start with the story again. I mean, you are starting a new life on land. Imagine you're a patch of cells, algae, or fern. Ferns are ancient organisms as well. Ferns, their reproductive systems are ancient. They don't have a vascular system. Ferns, that very good developed. I'm sorry, not ferns. The mosses, especially mosses, do not have a vascular system. Ferns somehow they have the basic, but they don't have flowers. Ferns, it's evolution. They have the vascular system. Ferns have, but they don't have the flowers. But mosses, they don't even have the vascular system. So they are like a carpet, growing on rocks, growing on logs, and on anything on the land, but it needs to be humid because they cannot withdraw water to higher elevations or to heights. Remember, sequoia trees, redwood trees, they are 300 feet tall. Even a fire engine cannot pump water higher than 10, 10 stories, 9 stories. I'm talking about a fire engine. So if you are living in a skyscraper, don't 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 rely on fire trucks much because that's how far they can pump the water. To my knowledge, I mean, back in the day, that's how I remember it. So it is a problem, this nitrogen deficiency. And then in my thesis work, the cells were dividing in a different manner. They had this pseudohyphal formation when you reduce the nitrogen concentrations in the media, as if they are trying to reach out to somewhere where they can find or forage nitrogen. And then this could be, we're talking about micromolar levels. Maybe like even yeast, baker's yeast, on like basic organism that grows on like rotting figs and grapes and etc. It has this tendency, in the absence of nitrogen, it grows towards it grows towards source of nitrogen, and this type of growth, this pseudohyphal growth stops at after actually 400 micromolars. It gets decreased, and at 1 millimolar you see nothing. I mean, it stops growing in that manner. But at 50 micromolar, wild baker's yeast, brewer's yeast, diploids, they show that pseudohyphal growth towards nitrogen if there is not much in the media. So even for fungi, this is a criteria, because nitrogen is necessary for growth. And then our laboratory at the time did some previous work and established that nitrogen is necessary, to control the major pathways in the cell, like TOR, for example, is a major controller. TOR, it's a protein kinase, and it regulates many functions in the cell. And in the presence of nitrogen, TOR regulates, regular, growth and development. But when it is, when the nitrogen is deficient, then TOR can, actually stop TOR can stop its suppression on some other metabolic ways, and let the cell try to find some nitrogen source. Normally in a regular cellular life, in a rich media, TOR is active, but sometimes, when there is not enough nitrogen, every cell, it's called, nutrient stress, or stress signalization. When there is not enough nitrogen, the cell can switch, certain metabolic pathways, towards finding sources of nitrogen. And so it is regulated in the cell, strongly regulated and conservatively regulated in all eukaryotes. That's what I'm trying to explain. In regular growth, nitrogen is necessary, and there is not enough nitrogen, that the cell, turns on other metabolic pathways, to reduce the activity, or or else, like switch to growth patterns and such. There are some, you can look it up in the medical libraries, and learn more about it. It's very complicated. I'm not gonna go into that detail, but on my blog I have listed the publications regarding the function of nitrogen and Tor, and actually the gene that I was working on, the SNF1 gene. It is named as sucrose non-fermenting. Uh, However, it was found to be active in the nitrogen pathway. So my research, was about that actually. How it was regulated. SNF1, protein kinase of the yeast was involved in nitrogen signaling, nitrogen cascade, and it was regulated, so I decided to, investigate, as part of the lab project, how it worked, and what kind of, protein kinases are involved, about the, SNF1 protein kinase, because SNF1 is a protein kinase as well. It phosphorylates other, elements, or other proteins. It's a conserved pathway. So that is the molecular part. I mean, that is very intricate and, complex. But in trees, so far, because I was investigating all these mushroom species, and you're talking about maitake, or the chicken of the woods. And now you may see in the woods that, like, they are giant, like huge, 10 pounds, 20 pounds of mushrooms, growing, or Fomitopsis species, how do they grow on a tree that is lacking nutrients? On the wood. Wood is not a nitrogen-rich material. Wood is not a nitrogen-rich material. So how do you see all this fungal growth, both on the tree, even on the ground? How does it start? And then when you start analyzing the food, for 16S ribosomes, for bacterial genome, when you start investigating all these ecosystems for bacterial genomes, you start to find the answers. I mean, you see bacteria, you see nitrogen-fixing bacteria in association with fungi, in association with the plant, especially in cold forests. They are living in the tissue of the plant, and providing growth to the tree, providing nitrogen to the tree, to the fungus, to the insects that are growing, or living on the tree, even in poplars, even in pine needles. Yes, it's been known that legumes and, beans and, many of those organisms have nodules, that have nitrogen fixing bacteria in the soil, though. That is a nitrogen fixing system in the soil, in the roots. That's why you don't have to fertilize them much, because they get their own nitrogen from the air with the help of those bacteria. So even like, if you keep investigating, and as I did so, you find the nitrogen fixing bacteria in honey, in the mouth of the larvae, in the gut of the larvae, in the crop of the honeybees, in the guts of the insects, in the guts of the earthworms. You know, they are everywhere. The nitrogen fixing bacteria is everywhere. You know, not just the roots of the legumes. They are in the guts of the insects. And then my project was, in my blog, it was the mycophagy, the consumption of, mycelium material by the insects, and dispersal of the, spores and or fungal matter with the bacteria, with the insects. So this opened another chapter actually in the project. Because now I see that the fungi on the trees need the nitrogen fixating bacteria, you know, to compensate for that growth. Imagine like huge clusters, those Maiteke fungi, or chicken of the woods, or other fungi that grows on trees. You need the nitrogen source to grow. I mean, it's just basic. The tree is full of carbon. The wood, the cellulose is full of glucose molecules, and glucose molecules are full of carbon. That is easy to find. It's in the atmosphere, and plants fix that. Plants do that already. But like incorporating atmospheric nitrogen into soil, into living bodies, that's a different story. That's the untold story. Fixing carbon is easy. It just and even if we are talking about like fixing climate, it's climate change, and we now we see all the atmospheric carbon is rising and rising and rising. You know, every year we're breaking a new record for atmospheric carbon. Nitrogen is one way to bury that carbon into the ground, into the living systems. And then without knowing the sources of those bacteria, where they live, how they live, do they form an association? Because they are very fragile bacteria. They depend on other organisms. Oftentimes they're gram-positives. They live in the guts of the bacteria. They live in the guts of the insects, earthworms. Oftentimes these are gut bacteria. And now they are doing all these research, isolating cultures, from the guts of the bacteria, and then doing nitrogen assays, and then see if there's any nitrogen fixing bacteria. They prepare a media like completely devoid of nitrogen, zero nitrogen, and then now they add the bacteria, and if it fixes nitrogen, you see some accumulation of nitrogen, even at micromolar level, with like 1, 2, 3, 10, even 10 micromolar is something, it's like talking about a small bacterial colony. These things matter in a tree, in the soil, in the decomposition of the wood. It's just like ocean. It's not different. We have whales to fertilize the oceans. I mean, if they are still out there. I mean, but when it comes to forests and all these carbon-rich ecosystems, nitrogen is scarce. It's not abundant, and then we have this bacteria that does the job. So investigating it is important, because you need nitrogen for cellular growth. It's just basic. And I see all these mushrooms growing on top of mountains, and I'm looking at it. There's just rocks. It's just one itty bitty plant, and then like king oysters. They're growing at high elevations in Turkey, in Europe, They give rise to these huge mushrooms, and then it doesn't make sense. Look, a small pile of like ferula roots. How does it get so big? There must be some association, with this bacteria. So I'm gonna go back to the beginning. This is actually it's a spore culture I made from the spore print. You rest the cap of the mushroom, on a piece of paper or even a clean, box, a plastic box, and then it just the folds, the spores, fold, and then even if it like clean water, like pure water, you can see mycelial mat after some time. It grows. The spores doesn't need anything extra to germinate. It's amazing. I mean, imagine it. This is even interesting. Maybe I mean, is there nitrogen bacteria is growing in it? We need to test, but the water is not cloudy. I mean, if this was like a super bacterial culture, it was a bacterial growth, and you wouldn't see this much clarity. It would be cloudy, you know. But what I do normally, like this is for example, this one is again a combination, a compost, for making mycelium, and then I inoculated this from the agar plate, and not from the liquid media. This is the solid system. You can like get a piece of fungal tissue, put it on the agar plate, PDA, that is mostly based on potato dextrose. But I'm gonna talk about one thing. You can also grow fungal mycelium with uh, with honey solution, 4% honey solution. And if you like boil it for 20 minutes to sterilize it, you know, uh, maybe it kills the yeast in it. Like I said, honey is not a sterile product. That's I have learned, and that is for you to learn actually. Honey, I was thinking, and everybody thinks honey is antibiotic, and it has antioxidant properties. Guess what? It is not. It is bacteriostatic. Actually, it has live bacteria in it. Maybe it has spores in it because it is coming from the mouth of the. It is coming from the gut of the the bee. I mean, it's just it's a live culture. I mean, it is dense. It doesn't permit bacteria to grow, but it doesn't mean it doesn't have any live bacteria in it. Maybe because the mycelium does very well in a honey culture. Like if you prepare a solution from 4% honey, even if you boil it for like 20 minutes, and then you inoculate the fungal tissue, it grows nicely, grows great. I mean, and if it is the right type of honey, I mean, if it is not like a fake one, you would get the right bacteria for the mycelium. That's what I'm trying to say. Even for mushroom growers, maybe this is one thing to consider. Honey and the bacteria in the honey, maybe that is necessary for fungal growth. Maybe maybe it is necessary to add some kind of considered as a probiotic. We need probiotics, for our gut systems. And what about fungi? We ignore it, and we try to grow it in a sterilized system. It's just one, monoculture, just like one culture of, oyster, mycelium. Is it? Does it happen that way in the nature? You know, how do you provide nitrogen? Normally, like regular growth, a medium for oyster mushroom, doesn't require high nitrogen. That's what their regular, growth pattern. It doesn't like nitrogen much. Maybe it gets it from the bacteria. Yes, you add some soy husks, and you have to incorporate some sort of nitrogen. But overall ratio, carbon nitrogen ratio is very specific when it comes to oyster cultivation. The high, there's the carbon nitrogen rate is very high in growth medium for oyster mushrooms and for many mushrooms, you know, because that's how it is in the nature. Like poplars, oyster mushrooms they grow on cottonwood, and poplars, and salix, willow trees. They're in the same family. And guess what? They are full of nitrogen bacteria as well, nitrogen fixing bacteria. I'm not talking about the bacteria that converts like nitrates to nitrates and other forms. That is something else. Conversion of nitrogen from one form to another is also important, but it's a different topic. I'm talking about fixing atmospheric nitrogen into usable form, into ammonia. That's how I did my work. With very lower concentrations of 50, macromolars of ammonium sulfate. That's how I worked. It was very precise. Like it was a very, very precise work. Like getting those concentrations is not easy. You have to be very careful. Like a little droplet of ammonia. That's it. I mean, if you if you don't, the, if you're not careful, I mean, it doesn't. You don't see that growth pattern. It gets like fuzzy. It gets into that fuzzy growth. The cells get elongated, and it's called like pseudo hyperfermentation. It's not a real fermentation, like the white rot fungi. So, and it's been almost 48 minutes. Like I said, so on my blog I have listed pretty much, uh, all the publications, that seems to be interesting in this field. Like the association between mosses and cyanobacteria, nitrogen fixing bacteria, the association between saprosylic bacteria and fungi, the association between nitrogen fixing bacteria and the fungi, and the association between the nitrogen fixating bacteria and the bugs and the bees and the other saprosylic bugs, especially coleopters, beetles. A lot of bugs rely on wood, dead wood. But like I said, dead wood is nothing. It's just full of carbon. I mean, that means energy, but for growth, it doesn't offer you much without nitrogen. Imagine like eating only bread. Even bread has some certain amount of nitrogen because it has gluten in it. It's a protein. Consider, can you survive on solely sugar, solely glucose? No, you can't. Nobody can. and why would you expect it from a fungi? That's the whole point. Can you grow? Like, can you survive solely on glucose, on sugar, solely on glucose? Nobody can. You need some nourishment. You need some food, protein, amino acids. And I think mostly this ammonia, generated by the nitrogen fixing bacteria is converted to glutamate, and the glutamate is an amino acid, and it's used for protein synthesis. That's that is the first step of the chain. Very interesting. For me, it was very interesting. I hope it is for you as well. And now we have all these papers and recent papers. I mean, when I look at a paper, I first look at the date, the the date of publication, and then there are recent papers, like 2019, 18 or 15 or 20, 21, 22. Many recent research is like backing this idea that forests are full of nitrogen fixing bacteria to generate soil, to generate life. I mean, to provide nourishment. It's not just in the roots of the legumes. It is everywhere. It is on the trees. It is on the bugs. It is on the bugs and the guts of the earthworms everywhere, in soil, on the trees, on the roots of the mosses, everywhere. Even in deserts, the desert truffles. It's just interesting. In the deserts, yes, there's one. There is one more way to generate actually usable nitrogen through the lightning and thunderstorms. With that, it's a energy demanding process. So during lightning and such, there is some amount of nitrogen turning into ammonia or some other forms. That gives chance to life in some areas, I guess. So rain contains some nitrogen, usable nitrogen, with the help of lightnings and etc. Is that enough? No. Well, that is something in an environment like deserts and other places, but it is one more way of generating usable nitrogen. And phosphorus, there are other elements, but the nitrogen is the most important one after carbon. Even oxygen. There are many organisms that can live without oxygen. And this process is actually oxygen free. It's interesting. The nitrogenase enzyme doesn't work in oxygen environment. It's special. So many people, that's why they didn't believe that. How could it happen? The trees are not suitable environments for nitrogen fixation because it's just aerated. There is lots of oxygen in the plant tissue. Is there? Maybe there are some pockets, and it needs to be studied more. And people are studying it and finding that there is nitrogen fixating activity going on in pine needles, in conifer trees, in poplar trees, in many trees. Especially the poplars. They are pioneer organisms. They grow on riverbeds and etc. Full of rocks. Have you seen a riverbed? I mean, it's just clean water flowing, and then you see all these sand and rocks. They love it. The willow trees, the poplar trees, they love the rivers. Where do they get the nitrogen? Again, they are fast growing trees. In many of the fast growing trees, It's like interesting. So I have added the many publications regarding nitrogen fixation because now I have found that many of the insects have this bacteria in their guts. They are already consuming this bacteria, you know, and they have it in their systems. And this bacteria is also present in the plant tissue, which is in association with the fungal tissue. So it's a triangle. I'm trying to like understand, and there's not it's actually just the basics. Everybody needs nitrogen. Fungi, bacteria, trees, and then interestingly, this nitrogen fixating bacteria seems to be friendly with every one of them. It's just one of the most friendliest organism on this planet. Interesting. It's a symbiosis, maybe. That remember that's how life started. It was a symbiote. The chlorophyll got inside another cell, and it became a plant cell. It's just mitochondria, same thing. It was a bacteria, maybe. That's how it is theorized. So, well, it seems to be the case still today. I mean, it started 800 million years ago. The need for carbon, the need for the energy, the need for nitrogen. The story goes on. So I am happy that my research actually is covering that part too. I wasn't thinking that at the beginning. Because nobody teaches that. The good thing about research, once you look up all these papers, and once you see that these bacteria exist everywhere, even in the bee larvae, in the guts of bee larvae, in the guts of bees, in the honey, you see this bacteria in the pine needles, in poplar trees, and in the willow trees, everywhere, in the mosses, roots of the mosses, everywhere. So the term, the cyprozytic, species, cyprozytic, organisms, actually, they also consume this bacteria because food is poor in terms of nitrogen, and oftentimes you see this bacteria on wood, which becomes soil at the end, which becomes forest at the end. Just maybe it's the keystone species. That's the term, the keystone species. Some species are so important. They bring the whole system together. Like imagine, like keystone is the single stone that holds the bridge together. Remember, like in the architecture, that's centerpiece stone that keeps the bridge together. Maybe the nitrogen fixing bacteria, and then bugs play a very important role in spreading these germs into flowers, because they are consuming, the bugs are consuming fungi, the bugs are consuming wood. It's just, and they have special organs that houses these bacteria, like the legumes, legumes keep it in their roots, and the bugs keep similar ways. They have special, the lobes or organs or sacs, or whatever, special areas that that they keep these bacteria growing, because like I said, it needs a special environment, especially it doesn't need any oxygen. It needs to be oxygen free. This process called nitrogen fixation happens in anaerobic environments. You need lots of energy, but free of oxygen. That's how it works. So so trees, helping with that. Gods are helping with that. The fungi is helping with that. So that's about it. So it's been an hour. So as I work on this issue, on this project, maybe I'll get to see more interesting points. But it is interesting already. To me, it isn't very interesting. Very interesting. That is the source of life. And nitrogen is very important. Thanks for listening. Have a good Sunday. Have a good week. It is already 2:15 a.m. Here in Turkey, Ayvalık, Turkey. Have a good holiday if you are into oceans and seas, and the seas are warming up, and enjoy the oceans, enjoy the seas. Please do not pollute the environment. Like I said, this environment is necessary for many organisms. So let's try to coexist. Let's try to coexist, and maybe we can find happiness that way. Thanks for listening again. Good day, good week. That's about